So recall that ecological communities are typically characterized by two data sets. One is a site by species data set, and the other is a site by environment data set. For the next 2 weeks, we'll be working with each of these. So last week's assignment was geared towards getting you thinking in a particularly analytical way, get you in a particular mindset, one of examining data and then graphing it. And the graphs allow you to detect things a bit more easily than you otherwise could, because we as a species are a very visually oriented species. And so detecting any potential patterns or problems can be easier when you graph it. And we're going to be following that kind of model throughout the rest of the semester. Today we're going to focus on working with site by species data. So once again, we'll be working with the BryceVeg.R dataset that contains vascular plant species data from Bryce Canyon National Park in south central Utah. Plants were surveyed in 160 circular plots that were 375 square meters in area. To put that into perspective, that's about the size of a basketball court. Each species of plant was identified. So we have species richness per plot, but in terms of abundance, individual plants were not counted. Instead, percent cover was estimated visually and used as a proxy of species abundance in two ways, both in terms of numbers of individuals per plot as well as abundance in terms of occurrence across plots. Excuse me. So each species in each plot was assigned to one of a series of mutually exclusive cover classes based upon its percentage cover. And you notice that these classes represent ranges, and the ranges are not the same sizes. So from 0 to 1% is not the same as the last class, 75 to 90%. Ranges were used because visual estimates of cover are by their very nature, very imprecise, and using a range allows you to deal with that imprecision a little bit better. But each of these categories, instead of being logically named, instead were given these numerical values that represent not actual numbers on the ground, but names. And so the actual data set consists of these nominal representations, these category names, rather than actual percent cover. So that's messy, but we'll just have to deal with it. Sometimes you don't get data that are precise or perfect. And so these are the data that we're going to be working with today, this site by species matrix. So as indicated in the notes, go ahead and open a new session in RStudio, set your working directory, and then load the library, the 6 packages that are indicated in the notes for today. And just like last time, you're going to read in the BryceVeg.R file as a data matrix that we'll call veg, and that's the code, the syntax below. For files that have the .r extension or a .txt extension, you can use the read.table function. If your data are in a CSV file, which is a comma delimited file, then the command is read.csv. If your data are as a D-based table or an X-based table, you use read.dbf. If your data are in an Excel format, as a .xls or XLSX, then I advise you to convert them to a CSV file. Just do a save as and save it as a CSV file in Excel. This will save you some hassles potentially later on. These are some examples of common questions that are asked about ecological communities, which we will answer with respect to the BryceVeg.R dataset. I've color-coded the questions here and in the online notes to make for easier cross-referencing. To answer those questions, however, we'll need to first transform the data to make them scaled similarly to each other, since our data represent intervals, recall, and those intervals are not the same size. There are many types of data transformations, each for a different purpose, and so I've put together a handy-dandy little table here of some, but by no means all, of the types of transformations that there are out there. So, for example, if you have a sparse matrix, that is one that is characterized by having lots of zeros, then there are a couple of types of transformations. One's called a chord, one's called a Hellinger, and so forth. If your data aren't normally distributed because there's a great skew, in other words, there's this sort of tail, then a log transformation is appropriate. If your data, instead of having a normal distribution, have a Poisson distribution, as is common with count data over time, then a square root transformation is appropriate, and so on and so forth. And so you can read this table and you can see the different types of transformations that there are out there, and you can see how much the log transformation is used. Log transformations help alleviate skew by rescaling values such that very large or very small values are brought closer to the mean. And so that's very useful if you have a lot of variation in a particular variable or within a particular site. So you can perform these transformations, and then in order to determine whether or not it's had the desired effect, you look at the residuals before and after a transformation. Looking at the residuals was something that you did in last week's assignment, and so you can just run that kind of code again. And what you were looking to see is if the residuals are smaller and no longer exhibit any kind of pattern, then you can conclude that your transformation was effective. Now, with the Bryce Veg data, we can't use any of these because the standard way of dealing with interval data is to take the midpoint of each one of the ranges, the intervals. And there is no midpoint transformation per se. And so in the online notes for today, I have indicated how you can do that for each one of the cover classes. So now we can get on with answering the questions. And the first of these asks about the distribution of species, getting at which ones are widespread, meaning found at lots of plots or sites, versus which ones are rare, found at only a few locations. And so to get at this, I've created an R object that I've called spc.pres, standing for species presence. I like to use short but descriptive names in creating R objects. Make sure not to have any spaces or symbols when you create a name. And then you use the apply function. This applies a command over an array. And in our case, what we're asking for is any cells that have values of veg that are greater than 0, in other words, presence, starting in column 2, since column 1 is the site names. And then the result is going to be a set of either presence, you've got a species there that is represented by veg greater than 0, or zeros, absence. And so that we just sum those presences to get at species occurrence by each plot. And then the results can be plotted. So by examining this, you can see, if you recall the veg matrix, there were 160 plots and there were 169 species total. So this figure shows that it took about really about 90 plots up here before all 169 species were encountered. So that's a goodly number. This pattern, this curvilinear pattern here, is fairly typical in terms of the distribution of species occurrences. It is highly skewed to the right, like this. We can correct for that through the log transformation. Remember that the log takes extreme values and brings them to the mean, and that has a linearizing effect. But what should we apply the log function to? Our objective in doing this transformation is try to clarify patterns, um, which is easier if you've got a linear kind of graph as opposed to a curvilinear graph that we had before. And so this command here, the log function is applied to the y-axis here, and it results in this graph. And so you can see how this log transformation linearizes the data. It keeps the data in their original form, their original scale. It just displays them differently. Now, if instead we had used this code where the log function is given to the actual species presence data, then you wind up with this. Notice the difference is in the y-axis. If we'd applied this log function to the species abundances, you get this graph. But in general, what we want to do is we want to keep the data in their original units for ease of interpretation. And so the first graph is preferable. So let's go back to it. It's this one here. Along the x-axis here is the number of species at or below a specific value, a y value here. And so, for example, approximately 100 out of 169 species were found at 10 or fewer plots. 130 species were found in 20 or fewer plots. So to answer our initial question, the answer is that most species were relatively rare. They were found at fewer than 10 sites. They were rare in occurrence. In other words, very few species were commonly distributed across the landscape. This is a very common species abundance pattern. We'll talk more about common patterns like this in a couple of weeks. So now we know that we have a lot of species that do not occur at most sites and a few species that do. And so that means that we can now ask a different question, this one. What's the mean cover of a species when it is actually present at a site? So this is asking about abundance in a different way. On the one hand, abundance can refer to the occurrence of individuals across sites, which is what the previous question addressed. This question in blue focuses on abundance measured as percent cover within each site. So once again, we use the apply function here to calculate the sums of the columns of the matrix veg, starting with column 2, the matrix that we transformed. And then we are dividing by the number of plots in which the species occurs. And when the result is plotted, you get this. You can see that this relationship is even more curvilinear, even more skewed than the previous plot of occurrence. And this means that most species only occur at very low abundances. Most are less than 5% cover. Only a few are abundant where they do occur. And by abundant, I mean above 10%. Still not a lot. In addition to the near universal pattern that most species are rare, few are common, both in terms of their distribution on the landscape, their occurrences, presences, as well as abundance within each site. Another truism is that widespread species tend to be more abundant than those species that are found in fewer locations. And this combines aspects of the previous two questions in terms of a new question, the one here. So, is the mean abundance of a species associated with the number of sites at which it occurs? So to answer this question, we need to plot the mean abundance of a species as a function of its occurrences or presences. And so that's done with this plot function here. In the plot function, the x-axis is listed first and then the y-axis here. And as you can see, this is plotting the relationship between species distribution or occurrence, presence, and species abundance. And this is the result that you get here. Each one of these dots is a species. And you can use the identify function to identify individual dots by their species names, their abbreviation codes. You can do that by clicking on each dot. And so go ahead and do that for some of the dots in each one of the corners as well as some of the ones in the middle. And then hit escape when you're done. The species that are on the right-hand side of the y-axis are the widespread ones, and you can see that most of these, in fact, have very low abundances. This is in contrast to what is more commonly seen, that widespread species tend to be abundant. That pattern is seen only in this species up here in the upper right here. This one is both widespread and abundant. Some of the high dots in the middle here are much less widespread, but they are relatively abundant where they are present. So, to get back to the question, is there a relationship between the number of plots in which a species occurs and its abundance? The answer is no, at least not for these data. So now let's get to richness and ask how many species are present in each plot. Once again, you'll use the apply function, like you did earlier, to assess species presence, absence, and then you sum that, which calculates the number of species in a plot site. The resulting graph of this shows that there are between 3 and 27 species per plot. Now recall that there were 169 species total. And so that means that even the plots with the most species represented only a fraction of the overall species richness for Bryce Canyon National Park. So the answer to our question is 3 to 27 species out of 169 per plot. Finally, we can ask whether the total abundance of vegetation is correlated with species richness of a plot. So this uses code very similar to before, first calculating the total cover of each plot and then representing the results in a graph. You will likely have to stretch the plot window in RStudio to see that the total cover per plot ranged from 3% to just over 100%. How can you get more than 100% cover? That's because each species was estimated individually and then those were summed. And so it's possible to get more than 100% cover in that way in a single plot. So then to answer our question about the relationship between total cover and the number of species per plot, you make a graph. And so you can see the result here, there isn't much of a pattern to this, indicating that there's not much of a relationship between the number of species present in a plot and the total vegetative cover of it. In other words, more diverse plots didn't also have greater abundance, but maybe this is because our data weren't true abundances. They were percent cover data rather than counts of individuals. So to answer our question, is there a relationship? No, not for these data. But maybe that's due to the nature of the data, the abundance data that we have as percent cover as opposed to the more traditional counts. So some of these truisms that I've been talking about today are more true if you're dealing with count data of individuals than if you're dealing with a proxy of abundance like we are here. So now you've seen some of the ways that a site by species matrix can be examined and how to interpret the resulting plots. For your assignment, you'll be doing some similar kinds of things and a few different things, asking and answering a few different kinds of questions using a different data set that comes in a different form, meaning a different arrangement. And it's this ground beetles habitat .csv file. And it's a data set of, as you can see, all of these beautiful beetles here. And with this data set, you'll be able to answer questions like, what site has the highest species richness? What species is the most widespread? And so forth. So it'll give you a little bit different kind of practice in dealing with a different form of data and in addressing some different kinds of questions with a site by species matrix. Next time we'll be working with site by environment data sets, and then finally after that we'll be able to get into the meat of the class in terms of dealing with analyses of ecological communities. Enjoy!